Weeds have always been a problem in agriculture. The newest challenges facing agriculture today are a changing climate and rapid development of resistance in weeds to many herbicides. While some weeds in a population may survive stresses from herbicides, competition, drought, and heat, the plants we depend on for our survival may be less tolerant and struggling. One weed that's particularly resilient and of great concern to the farmer is Amaranthus palmeri, better known as pigweed. It has developed an amazing ability to resist many of the chemicals used to control it. Pigweed can reach as tall as 8 to 10 feet, often dwarfing the intended crop, with a single mature female plant capable of releasing up to 500,000 seeds. It's extremely challenging for growers to harvest their crops without further spreading the weed, either by inadvertently transporting from field to field in grain and on farm equipment, or carried by animal and bird delivered in droppings on their journeys. In fields heavily infested, harvesting becomes impossible, resulting in loss of income, as harvesting can cause excessive damage to field equipment. In many cases, farmers choose to skip over the affected areas, leaving behind crops and cutting into their income. The versatility of pigweed drove ARS's Dr. William Mullen and a handful of scientists to find a new way of combating this problem. Instead of just adding more chemicals as a control solution, these scientists hypothesized the idea of looking for a natural solution. They turned to the pigweed itself to understand why it was able to adapt so well. After 5 years of study, Dr. Mullen and his primary collaborator from Clemson University, Dr. Christopher Saski, found something new, or maybe something that's been there all along and we just now are discovering it. Most importantly, it could be the key to understanding cell adaptation on a larger scale. The mechanism by which pigweed survives was a natural molecular mechanism, completely designed and selected for by pigweed itself. It represents a new mechanism of transmission of genetic information previously unknown at this scale. With this discovery, we may someday be able to control pigweed and other nuisance weeds and invasive plants. More importantly, we may be able to improve desired crops, giving them the same ability to thrive in a changing environment and possibly even resist threats from new invasive insects and pathogens. Who knows where we'll go from here?